Welcome to Basic Science Series. I'm your host, Lokendra Kumar, and today I will be designing this structure. After this video is done, you will be seeing this structure, and while we are designing the structure, we will also discuss each component, what is the role of these domains, why they are important, what is FAB, FC, and all. So basically, you will be knowing how to design antibody structure, and you will also be knowing what are antibodies and why they are important. So let's get started. Today, we will be working on antibody structure. It's also known as immunoglobulin. So what I will be doing in this case is I will try to design the structure of antibody. At the same time, I will also discuss the functions as well as describe the various components of antibody, and we'll see how much we can discuss about the structure of antibody. So in this video, basically, you will know how to design the antibody structure from scratch, and what are antibodies, why they are so important. We'll discuss some of the important points. So it will be a long video, obviously, but it will be more like the detailed and relaxing content where you will be understanding the structure. So do not expect this to be a very quick and fast video. You can also follow through if you want to design the structure. I'm basically using basic shapes in Microsoft PowerPoint, so you can follow with me. I'm using this area because then the structure is quite visible and it's really easy to see. So let's see. I have a basic layout in my mind, and you have seen the antibody structure also in many books. Okay, so we'll start with making the chains. As you all know, antibodies, they contain the chains, heavy chains and light chains. So let's see which shape I can use. Probably this one. So this is a typical shape. Okay, so I'm thinking to use this typical shape for all the chains. So I think probably this is good enough, right? Okay. You can also work with grid. All right, so let's color it differently. So I think I can use the gradient color fit. So in this case, probably up to you which kind of like gradient color style you like. Okay, so that is fine. All right. Usually they have different domains. The structure of antibody is different, right? It's not like this. It's the schematic that you have to understand. Okay, so if I take this as one part and just copy and make it another one, so that's the one chain. Probably I want to use a different color for this one. I don't like it because it's matching to my background. So no for that. Maybe this one is quite nice. Yes. Okay. So outline, I would say we'll go with the dark black color outline. You can also use different shades of gray basically to make the structure. So this is one, right? You can just simply copy and then it becomes the second chain. Interesting. Now if I want to make another one, so I will just probably move it like this and then move to the other side, which is here. So these are nothing but the chains of our antibody molecule. Okay. Right. Let's make them a little bit closer. These two as well. Okay, now we also need the FC fragment. I'll tell you what are the FC fragment, and then this is the FC fragment. It's also globular, so basically we need to have at least 2 chains down there. So basically how many chains you have? So you have 1 2 3 4 5 6, and they are combined, so you have basically one heavy chain and this is the light chain. So let's change the color theme of these, and in one go you can basically change, and it's very, very easy to change the shape. So I think this is much better. Okay, I have to zoom in, zoom out, because some of the details we have to see how we're going to put them. One thing I definitely need to do is just make the gap in between these chains, because we will be adding a specific bond. Okay, so this looks better. So now you have also a significant gap in between these chains. All right, so now I need to connect these two. You can use different tools. I will simply use the line function, and so basically this weekly line function here. Correct. So you can see, you just drag down by mouse clicks, you will have something like this here. I will probably change the color of this, so I'll go to the shape outline and make it green, because I'm using heavy chain as green. I'll increase the size so weight can be increased. Now it's looking much better. Okay, I think it's good if it is like this. All right, okay. There you go. So what I'm trying to do is basically connect these together so that I can give the justification that these chains are basically, they are heavy chains. So what I'm doing is basically I want to hide this region, so I will just cut it and again paste it. So you can see you get the illustration the way you want. So now the heavy chain is basically ready. Correct? Okay, this looks much better. Why is this? Okay, anyways. Okay, so probably I need to write something, then this option will go away. Use dimension H theme. Okay, next is, now they are connected, and they are also connected with the disulfide bond. So that is going to be challenging. If I want to make the disulfide bond, how I'm going to make the disulfide bond in this case? All right, what I can do is basically, can I use this? I don't know. I think I can use the line function again and just fill it out with different colors. So probably that will be much easier for me. Let's see. I'm just trying quite a few things here, but it's not straight, so it's not that nice looking. So probably what I need to do is basically turn it to this side. Now you can see this is in front of that, and then probably let's use the shape function, the line function. Just simply we can start one line from here to here and another click from here. Okay. You'll see what I'm going to do next is basically, now you can fill it with the same color. So let's fill it with the same color. So you can see the connection is there. Correct. And you can like, let's make it a little bit transparent. So you can go to more fill colors, increase the transparency, and then let's probably, what we can do is outline, remove no outline. Look, so this looks much better, right? So you can just copy this one and you have the second disulfide bond here, because usually you have two disulfide bonds in case of these two heavy chains. Okay, perfect. Now in addition to this, to make it more better, so we can do this S and S, right? So that indicates the disulfide bond. Disulfide bond is nothing but you have two sulfate molecules. I usually prefer to use Arial font, so that is what I'm going to do, and then maybe recenter it. Is it inside this? Okay, so this is something I don't want. So I think I've also changed its position as well. Anyways, okay, so one disulfide bond here. Let's zoom out. Second one here. Okay, not that. Okay, so if I do this again, it came with the structure. So what you can do is just remove the fill and put it directly over here. Now you can obviously adjust the position so it's much better and clear. Okay, so now what we have done is basically we constructed the heavy chains, and then we connected these heavy chains with the disulfide bond. And this region, which is curved region, you can see is the hinge region. So let me again tell you a little bit about the antibodies. Antibodies are nothing but, also known as immunoglobulin. They are protein molecules produced by B cells of the immune system in response to the foreign molecules such as bacteria, virus, and other pathogens. Now, basic structure of an antibody molecule consists of 4 polypeptide chains, 2 heavy chains and 2 light chains. So you have 2 heavy chains, 1, 2, and 2 light chains, 1 and 2. Correct? And what is next? They are linked together by disulfide bonds. Each chain has a variable region and a constant region. Now we have to also connect these two together because they are also connected with the disulfide bond, and these are already connected. So what we can do is basically, since we already have that structure, I think I will just simply use this. Now if I move this like this, I can simply fill the area with some other colors. Should I do that? Like this probably? Correct. Okay, so they are now connected. So this disulfide bond is between heavy chain and the light chain. The green one is between the heavy chains. So that is important. Okay, so what I can do to actually hide this artifact, we don't want that. We have to actually move this particular thing to the background. So what I can do is select all these structures and then just cut them out and again paste them. So you can clearly see now it's looking much better. Okay, so we should also start labeling these things, right? Otherwise we'll be confused, right? So heavy chains and light chains. So I will write it down, HEAVY, and then H chain, since this is the one chain. And as I told you, for structures, I basically use Arial font. Looks way better than other fonts. Should we revolve all these things all around? So I think, like, what should be the size of this? I think this is fine, probably. I'm just comparing the size to the sulfate bonds. Okay, heavy chains. Now it's time to label the heavy chain. So probably that is the heavy chain here. So let's do this. Let's keep it closer. Now I want to move this. This is the heavy chain, correct? Then the second one is also heavy chain, okay? And we can also write the domain name, in this case C. Okay, so we should not write that in this way. What else? Sometimes you also have carbohydrate molecules attached to the heavy chain. So let's, you have lots of options of different types. I can use maybe this one. Is that okay? So let's use this one and place it right over here, and I want to use another color, probably magenta, or let's use this one. Seems like, okay, no outline. All right. So this also goes to this. Now we can do this like and attach them. If you are able to attach, that's very good. If not, you just cut them out and then replace them. So these are the sugar molecules. So let's also label them carbohydrate. I will collect the spellings if it is wrong. So now I'm making another label so that I can label this. Honestly, these are a little bit thinner, so I'm going to make them visible and then change the color. So here you have it. So this is the heavy chain, this is the carbohydrate. Now we have to label this as the light chain. So that is nothing but L here. Here you have light chain. I'll just copy this from here and then place it like this. You can do whatever you want. It should be organized, and labeling the way you want to do should be understanding to the readers. That's important. You can also use quite few options. So basically you can use alignment options and so on. Right now I'm just making the structure so that you know how to make the structures. Now what is next? So since we have called them different regions for heavy and light chain, so we can also give them a specific name. So for first one, I'll just say VL, variable L. This indicates that this is the light chain variable region. This is the light chain, but this is constant region. Okay, I'll explain what I mean by that, but before that I have to change it for heavy chains too. So this is the constant region for heavy chain, and then the same way this is the variable region. Now, what is the logic behind this? In antibody structure, you will see that one is the variable region, another one is basically the non-variable or the constant region. This is important. So if I explain it in a different way, hopefully you will be able to understand that the amino acid sequence of the variable region of antibody chain basically determines its specificity for a particular antigen. The variable region, it contains hypervariable regions, also known as complementarity-determining regions, CDRs, which directly interacts with the antigen. So basically what is happening here, every antibody will have different affinity against different type of antigen, especially when you have polyclonal serum. Different antibodies will have different affinity against different types of epitopes. So this region will vary a lot. Correct? So this region will be changing its shape depending upon what kind of antigen it will encounter. So the structure of this will be different. This pocket will be entirely different. Okay, that is why you have the different types of names for these regions, right? So I'll move same thing here. Oops. We have to flip them up, right? This is very, very important to understand that which one is variable, which one is constant. The same way, let me just copy this one. All right, so this is heavy chain. This is the constant region for the heavy chain, and this is the constant region for the light chain. Perfect. Looks much, much better. And then what are these then? What are these? Because they are again the constant region of the heavy chains, we need to divide these regions with different numbers. So I'll call it as 1, CH1, correct? And then I can move them down here. So this one is 2. Look how easy this is to understand the structure of antibody by just simply drawing the structure and understanding every component of it, right? So 1, 2, 3, and then you have this is again 2 and this is 3. Now how you will be differentiating between these two? So that is the question, which is again is very, because they are same, right? This entire structure is same, so you have the same names for these domains. Now they are not written like this, so basically you have to decrease the size of these to make it correct. Let's decrease the size of this. How much? 9? Okay, 9 is better. Here 9. So this is how you basically label them, right? 9. And then in this case H will be smaller, and then in this case again we have to reduce the size, so you have to bear with me. I already told you that it will take some time, but you know, hard work pays off. So you can see, all right, 9, and then again for this one, all right, 9. Should I just type it? Yes, this is much easier. I just type 9. There's no need to just decrease the size by just again clicking, re-clicking. Okay, so this is much better. This is much better. So you can see this is the structure of the antibody molecule. Now I have to show actually the antigen binding site. So probably I should make another structure right here, and then what should I do in this case is basically let's keep the outline same, and I'll just keep the transparency really low so that we can see the structure. Not for this one, but for the fill. All right, something like this, much better. Okay, so I'm gonna just select it, move it on the top. Okay, I have selected everything, so that I don't want to do. I have to just select these, and then it comes on the top. So you can see this is your antigen bound to the antigen binding site. So I'll call it as antigen and move this guy to right over here with one label, and this is your label, and this is how antigen binds. You know, it's schematic, I already told you. So the same thing is going to happen here. So just copied it, no rocket science behind it, and just copy it, and there you have it, second antigen. Okay, so let's look at our structure. Look at this. This is something you will see in very, very high quality textbooks. The antibody structure. Let's see if we have covered everything. There are a few things, and we haven't talked about the regions. So you have FAB region, antibody binding region, which is basically this particular region, and FC region is the region where you have these CH2 and CH3 parts of the antibody. And what is this? This is basically nothing but the hinge region, which is a little bit flexible. So disulfide bond 1, 2, 3, 4 disulfide bonds are here. And we can also, you know, there are so many structures that are available. I just thought I will draw this one. So you can see this is also looking good, right? And sometimes you will see the structures, they are different. Sometimes you will see they are different type, but overall basic function as well as the description remains the same. FAB fragment, FC fragment should be labeled. This one, let's do that. You know, you've spent so much time, we should also do that so that it's clear. Look, you can basically change the structure of this one like this. You have these yellow circles you can use. Okay, let me increase the weight. Fantastic. This looks much better. I think I just stepped a little bit down for this. It should go a little bit higher. This should be like this. Okay. Now this one is the FAB region, and probably I want to write it with the bold letters. FAB regions. So let's, obviously I need to increase the size so it's visible. Should be bolded. I don't know. Let's decide. This is our antibody, so we can play with it and change different colors and all if we want to. Anyway, so this is FAB, this is FC. FC region of the antibodies basically binds with the cells, and then these are exposed to outside to bind with the antigen. An antigen can be any foreign substance, and we already talked about it. Next, is there anything else? Yes, we were talking about the constant region. So constant region of the antibody or the heavy chain determines the antibody's effector functions such as complement fixation, antibody-dependent cellular cytotoxicity, opsonization. There are so many functions because this part is basically sitting on the cell. This is very, very important to understand. So we can also show this if we want to. Okay, let's just try that and see if we can show this. So basically we can just simply, is that a cell? Definitely not, right? But if we expand this like this, it's gonna look like a little bit surface. I'm not happy with this structure, so let me just draw it. Okay, so usually surface, they are not straight, so they are a little bit wiggly, and then it will go like this. So membranes, they are like this. Okay, we can fill it up. Wow, that looks much better. You have gradient options. We don't want to insert the images, okay, because we create our own images. That is very, very important. You need to create your own images. All right, looks much better. You want to increase the width so that it becomes like membrane shaped. You can also put a little bit of darker yellow color on this, and antibody structure can go on top so that it's complete. There you have it. Look at that. So this probably is the cell here, and I will not make another structure inside the cell. You just have to imagine this is the cell surface, antibodies bound with the heavy chain Fc region, and then light chain is exposed to have interaction with the antigen. So you can see antigen is here, antigen is floating, and then it has the ability to stick with this part, and now it is fixed. Now it's been neutralized. That's the whole purpose of the antibody molecules. Now antibodies, they can exist in different forms: monomers, dimers, pentamers, depending upon their isotypes. We are not going to discuss the isotypes here, but yes, we will move on to different isotypes. We will try to understand their function. We'll try to understand the structure of those isotypes. It's really, really interesting and really, really important as well. Right, so if we summarize this, antibody is basically consisting of 4 polypeptide chains, 2 heavy chains, 2 light chains with variable regions, and forms the antigen binding site and have the constant region that determines the class and the effector function. So I hope the video was helpful for you to understand the antibody, understand its function, and also know how to design a high quality illustration of an antibody molecule. Honestly, I really liked it. I don't know. You can also put your comments, and you can also tell me if the structure is nice, if there is any left out thing that I need to add. Please feel free to put your comments, and then I'll definitely try to answer all those comments. Till then, thank you and take care.